All right, everybody. So we're going to take a look now at mitosis, but we're going to look at it from the angle of that mitotic spindle apparatus. So in class, we hit a freeze-framed view of prophase, metaphase, anaphase, telophase, and what's going on in the big picture, explaining what happens with a mitotic spindle. But let's really zero in on that with a deeper look, a deep dive into those stages with the mitotic spindle apparatus in mind, okay? So let's start here. During prophase, assembly of the spindle microtubules begins in, and this is one of those organelles that we met in chapter 7. So this is the centrosome. All right, so now we got a whole bunch of C words here in chapter 7 and 8. We have centrosome, we have centrioles, centromere, and you got to get those straight in your head. But the microtubules begin in the centrosome, the microtubule organizing center. That's its nickname. Some people even call it the MTOC, microtubule organizing center. Okay, so get that. Try to allow that your brain to absorb that in. Okay. Now, as far as that MTOC, your centrosome, it's this whole big thing here. Okay. So this whole region here with the centrioles in the middle, you have this ring here, and then you have these microtubule rays coming out of it. This whole thing, that's your centrosome. Now, the mitotic spindle apparatus, if we use that phrase, we're basically talking about the centrosome and what comes out of it, which is a lot of microtubules that control the chromosomes and their movement during mitosis. Okay? So all those little rays that you see starting to come out of the centrosome there, about half of them are going to connect onto chromosomes, as we said the last day we were together in lecture. And they're going to allow those chromosomes to be moved to very specific places within the cell during this process of mitosis. Now, we need 2 centrosomes during mitosis for this to happen in animal cells. So you start every cell with 1, but you got to have 2, just like we've been saying. You got to double up everything. You got to double up the organelles. You got to double up the volume of the cell. You got to double up the DNA. Well, you also have to double up the centrosomes, because what's going to happen is we need both of them in place, because one of them is going to move to the opposite end of the cell. It's going to migrate around to the other side. And as we can see here in this picture, you have centrosome 1 here, centrosome 2 here. They're on completely opposite ends of the cell at this point in mitosis. That's why you got to have 2. All right. Now, plant cells are a little bit different in that they lack some of this machinery. But since we haven't done a deep dive into the plant kingdom yet, that's more of a semester 2 thing, we'll just note some key differences between animals and, say, plant cells. And one of them is that they don't have all of the same machinery like centrosomes and centrioles, but we're going to be kind of focused on eukaryotic animal cells as we learn mitosis. Okay, now those centrosomes are in the form of what are called an aster. Okay, sometimes they're referred to as asters. Asters have a central hub, and they have radial arrays around them, okay? And we use actually the term aster quite a bit in botany as we describe plants, because it has the central hub, and then you have the petals coming out in all directions like rays. So they extend from the centrioles outward in all directions. So once you start adding the asters themselves and the microtubules that are currently growing out of them, you're basically now making a spindle apparatus. So the 3 things that any spindle apparatus will have during mitosis is the centrosomes, and I'll put a 2 there. There are 2 centrioles in each centrosome. If you want, we could even count them. 1, 2, 3, and 4. There they are. And of course, all of these rays coming out, these are proteins called microtubules. And we met these also in chapter 7 when we had our cell chapter. We said they were part of a cytoskeleton. We said there are microtubules and microfilaments, and not only did they provide structure for the cell, but they also made like an internal highway system so things could move from one place to another. We said some of them would strap down to the cell membrane and allow the cell membrane to blob out and around something that it wanted to pull into the cell during endocytosis. So centrosomes, centrioles, and microtubules make up the spindle apparatus. Now, those microtubules, again, they're going to be connecting onto something very, very important, which are the chromosomes, as we can see in the picture. Some of those yellow microtubules are clearly connecting onto the chromosomes that are purple with the black little spots. Now remember, the last time we were talking in lecture, outside of the little narrow waist of an X-shaped chromosome, we said there were proteins called kinetochores. The kinetochores were like little handles that the microtubules could grab onto so they could move those chromosomes to where they wanted to move them to. So those little kinetochores are here. You can see them in black here and here, provide a handle. You can see those microtubules connecting right to them. Okay? And where they're going with these chromosomes is to that imaginary line down the middle of the cell, almost like the Earth's equator, right? That splits the northern hemisphere from the southern hemisphere. But in biology, we call that the metaphase plate. So the metaphase plate goes down the middle. That's where all the chromosomes are. That's where they all essentially get pushed to or pulled to from those kinetochore fibers. And this is the most distinguishable stage of mitosis, because when you look under a microscope, they're all right down the middle. Now, in the next stage of mitosis, called anaphase, we said that the big moment was when those sister chromatids of the chromosomes break apart from each other. So in anaphase, your sister chromatids separate. And they begin walking back to each of the poles. A long time ago, we used to think that they were being pulled to the poles during anaphase because both of the halves of the sister chromatid would kind of, it would almost like they were on a string being pulled up, because the legs would kind of point downward, almost like they were being pulled up. But we found out in later years that that wasn't happening at all. Instead, it was something much more interesting. We had, here is the kinetochore in this teal-colored circle. And inside the kinetochore, we have these motor proteins, like these little red feet. And through chemical interactions, we found out they actually walk. So the kinetochore walks the chromosomes back to their respective end inside the cell. Maybe you're headed towards the top pole, maybe you're headed towards the bottom pole. And what's even more cool about it is as it walks up that microtubule, it disintegrates and the pieces come apart and get released as kind of like spare parts for the cell to use for something else. It would be like if you were walking down the sidewalk and you looked behind you and the sidewalk was disintegrating behind you as you walked forward. That's what's going on here with these chromosomes as they move towards the pole. Now, this particular setup and arrangement here, this is a kinetochore microtubule. Here's the yellow microtubule. It's connecting to the teal-colored kinetochore, which attaches to the chromosomes. But if you look down here, it says, meanwhile, non-kinetochore microtubules. What the heck is a non-kinetochore microtubule? Well, it's one that doesn't connect to the kinetochores. Instead, these microtubules don't have a chromosome between them. They push against each other, one from each side, and what they do is they elongate the cell. Sorry about the white, doesn't always come across the screen so easy to see. Elongate the cell. So as we saw in our activity in some of our previous talks, that cell goes from fairly round to stretching out, a little bit oval, stretching out a little bit further, a little bit further, a little bit further, until it's really oval and has a pretty long vertical axis through it. So it needs to do that so it can break into two cells. So here's an example right here. You can see that the microtubules that connect to the chromosomes here are green, and then these red ones that still hang out in the middle, notice how there's no chromosome at the end. They're just pushing against each other, making this side of the cell go this way and this side of the cell go that way, elongating it outward. Okay, so then the chromosomal material reaches each pole, and what we have in the last stage of mitosis, telophase, is genetically identical daughter nuclei. Remember, because we duplicated the DNA during S phase, what reaches each pole now is identical copies of the DNA. And you can see during telophase, some of the things that happen is we start to reform our nuclear membrane back. We start to get our other things back, like our nucleolus, our ribosome producing center. We also start to see, we call in biology, when it does a V like this, it's called an invagination. So it's starting to fold inward in the cytoplasm, which is going to bring up our next big step of cytokinesis, because we're still technically one cell. We have one membrane around it. This is what it looks like in real life here. You can see right there. You see that folding inward of the cell membrane as it's pushed about as far sideways as it can. And this process, where ultimately it's going to keep going in that direction until one becomes two, we call that cleavage. Okay, if you think of like a meat cleaver, it's a big knife that cuts big amounts of meat from one big piece into two pieces, right? So cleavage is cutting something in half. And the cleavage furrow is this little V-like shape that you can see right here. And that's going to get deeper and deeper into the middle until it finally separates one into two. Some people like to think of it like a drawstring around, like, you know, since cells are fairly fluid, if you were to tighten a drawstring around the two of them, like you can see in this picture, eventually it would get so tight that it would snap it into two, and the two would pop away from each other. Okay, this is what it looks like here from electron microscope when a cell is dividing into two. Here's that cleavage furrow. You can imagine it's kind of like a V right there, and it's going to continue to do so until one becomes two. And most cells will do it just like this. Now, there is a couple of exceptions we should talk about here. Some types of cells can go through the mitosis process. And again, mitosis is the dividing of the nucleus. And it won't go through cytokinesis. So the logical outcome of that would be more than one nucleus in a cell? And the answer is yes. Okay, you can have multinucleated cells. In cells that have a high amount of demand, like muscle cells that need to contract and expand and then relax. Your heart cells, for example, they have to beat 24/7 for as many years as we're alive. Your skeletal muscles that provide so much movement of our big human species organism that we are, and that takes a lot of control, a lot of coordination. So you can have multinucleated cells inside of organisms, and for our species, muscle cells are a great example of that. And then lastly, let's just kind of say a few words about plant cells. So earlier we said they lack some, there are some subtle differences between animals and plant cells. We just don't really have the time to fully dive deep into plants right now. But what we can say is they do lack some things like centrioles and centrosomes. They're able to get it done without it. Probably the biggest difference is that you don't go through cytokinesis in the same way. When you split the cytoplasm, it's not from a cleavage furrow. Instead, we build a wall down the middle. Okay, it's called a cell plate. So, if you guys remember the Golgi apparatus, we remember that vesicles kind of blob and float into it, and they get worked on in some special way, transformed or perhaps changed, and then a new vesicle will come out the other side and will head towards the cell membrane or build something inside the cell. Well, these here, they build this structure right here in the middle called the cell plate. It's literally building a wall between the two sides. The nuclei have already done their mitosis bit right here, but now at the very end, they're building this central wall here called the cell plate. And a little bit later, what you see is two new cell walls will form from that cell plate. And as you can see, it cut the volume in half from this original cell. Something in the center called the middle lamella is just kind of a internal layer between those two cell walls. And then the plasma membrane is right here strapped up against this cell wall, as long as this plant has adequate water content, hydration. Okay? So just a little key difference there with plant cells.